Welcome to part 4 of our discussion of cells. We're going to be discussing transcription and translation. It is very important that you understand the difference between these two processes. You may have something that increases transcription but not translation, or you may have something that increases translation but not transcription. As you go forward in biology and human anatomy and physiology, there will be much emphasis on the molecular mechanisms of cellular changes, and understanding the basics of transcription and translation will serve you well. First of all, we need to clarify the difference between transcription and translation. These two processes allow genetic control of cell actions through protein synthesis. The DNA is what directs the synthesis of all cell proteins. This includes enzymes that direct the synthesis of non-proteins. Things like lipids or glycogen are stored carbohydrate. Different cells are going to synthesize different proteins. And this is dependent upon differing gene activation. So we have different genes active in our liver versus our brain. There are some genes that are active in all our cells, but there are many that are active only in some of our cells. So when we go from DNA to RNA, mRNA, messenger RNA, that process is called transcription. We directly copy the DNA and then we process that pre-mRNA that's created as a copy of the DNA. Once we have processed it and we've edited it and recombined it, it becomes mRNA and leaves the nucleus through nuclear pores. Once the mRNA is out in the cytosol, the small and large subunits of the ribosome will attach to the start sequence and begin the process of translation. Translation is turning mRNA into a polypeptide, the beginning of a protein. The genetic code, which you learned about in your previous biology classes, is a system that enables 4 nucleotides, A, T, G, and C, to code for all 20 of the amino acids. This is based on a 3-letter language. We call this the base triplet. It's found on the DNA molecule. An example of this would be TAC, so 3 different nucleotides in a certain sequence. These are the nucleotides that stand for 1 amino acid. The codon is the mirror image sequence of the nucleotides found on the mRNA. Remember, A and T always pair, C and G always pair, and in RNA we replace our T with a U. That means that if we have a T in our DNA, we are going to have a U in our mRNA. There are 64 possible codons, that is 4 nucleotide bases raised to the 3rd power, because there's 3 letters in each codon. And because there's only 20 amino acids, you find that 2 to 3 codons often represent the same amino acid. We always have the same start codon, which is going to attract our small and large ribosomal subunits to the mRNA and tell those subunits where to start the protein. Our start codon is AUG. We have 3 stop codons that are all very similar. They all start with U. One is UAG, one is UGA, and the other is UAA. Whenever the ribosomal subunit, the small and large subunits that are attached and translating that protein, they get to one of those stop codons. That's the signal that tells them to let go and stop translating. So back to transcription, what happens in the DNA. There's a video available on Canvas that you can watch at your leisure if you would like to see this process in action in real time. Transcription is the process of copying instructions from DNA to RNA. We form messenger RNA, mRNA, next to the activated gene. The RNA polymerase binds to the DNA. We know that RNA polymerase is an enzyme because it ends in the letters A, S, E. That enzyme opens up the DNA helix and transcribes the bases from one strand of DNA into the pre-mRNA. Remember, we're going to edit and recombine that mRNA before we send it out of the nucleus. The polymerase then rewinds the DNA helix. So it pops it open, copies it, and then puts it back together. Pre-mRNA is unfinished. We have to take out nonsense portions. We call those introns. Those nonsense portions are going to be removed by certain enzymes within the nucleus. Those enzymes then take the exons, and I remember that the introns stay in the nucleus, the exons exit the nucleus. The exons are the sense part of the pre-mRNA, the part that we actually want to turn into proteins. We reconnect those sense portions, and the mRNA is now finished and can exit the nucleus. It's going to migrate through the nuclear pore to the cytoplasm. The steps in translation of mRNA. So translation is when we go from mRNA to a protein. Translation is the process which converts the alphabet of nucleotides into a sequence of amino acids to create a specific protein. So translation involves three major steps. The initiation. This is when we form the ribosome in the cytosol or on the rough endoplasmic reticulum. The small subunit attaches to the mRNA leader sequence, that's that start sequence. The large subunit joins and pulls the mRNA along as it reads the start codon. So the start codon is where the small and large subunits come together and protein synthesis begins. Remember that start codon is AUG. The small subunit binds the activated tRNA with the corresponding anticodon. So we have codons that are located on the mRNA and anticodons which are located on the tRNA. We're going to match those two up, and that determines what amino acid is attached. The large subunit forms the peptide bond between amino acids. You can see in this frame from the focus figure on transcription and translation in Chapter 3 that we transcribe our DNA into mRNA. Our mRNA leaves the nucleus and travels to a ribosome. The small and large subunits bind to the mRNA strand, and they start with the codon AUG. That's our start codon. Our start codon is going to have an anticodon that matches up with it. Because A pairs with U pairs with A, and C and G pair, the anticodon for our start codon is UAC. So we're going to look for the tRNA that has the UAC anticodon to start off our protein. That tRNA with that specific anticodon always binds the amino acid methionine. So MET is the abbreviation for that. So that's always our first amino acid in our protein chain. Once that comes in and binds, the next step is to bring in the next tRNA molecule bearing the correct anticodon for our next codon. It's pretty much like matching up puzzle pieces. This process, going from mRNA to amino acids, is called translation. One hint to remember transcription and translation is that a court reporter transcribes what's being verbally said into the same language but written down. Translation is going from one language to another. So transcription, the language is nucleotides. During translation, we translate from those nucleotides to amino acids. So it's a different language. It's the amino acid language instead of the nucleotide language. So that might help you remember the difference between translation and transcription. So the next step in translation is going to be elongation. That's when we take our amino acid chain and it gets longer and longer and longer. So the ribosome reads the next codon, attaches the correct tRNA with the correct anticodon, brings in the next amino acid, and the large subunit will form that peptide bond between those two amino acids, and the chain just gets longer and longer and longer. Termination is when we reach the stop codon. The process is halted when we reach the stop codon. The stop codon signals the polypeptide, that big chain of amino acids, to be released, and the ribosome will let go or dissociate into its two subunits. So the small subunit and the large subunit will float away and find another mRNA to translate. This electron micrograph here shows multiple ribosomes along one mRNA molecule. So I'm going to highlight that mRNA molecule. It's basically like a ribbon that goes through all of these ribosomes. So there are more than one ribosome on one mRNA molecule. And each of these ribosomes is producing a peptide chain. So more than one peptide is being produced along this one molecule of mRNA. That's what allows us to make proteins so quickly. So tRNA, transfer RNA, has some unique features. It has a unique three-dimensional shape, and that three-dimensional shape includes an amino acid at one end and an anticodon at the other end. This unique shape is what allows it to match up with the codons and deliver the correct amino acid. In order to attach the amino acid, we have to activate the tRNA. So activation by ATP, adenosine triphosphate, binds the specific amino acid to the tRNA. This energy that we release from ATP provides the necessary energy to join the amino acid to the growing protein molecule, that growing polypeptide chain. What we do is when we attach the amino acid to the tRNA, we store that energy away, and then we can use that energy to attach the amino acid to the next amino acid in the chain. Remember, the anticodon always binds to the complementary codon of the mRNA, like a big puzzle. So I would highly encourage you to review protein synthesis during translation, a portion of Focus Figure 3.4. Go ahead and read through that and understand the steps in the process, how translation begins, how it progresses, and how it ends. So the three main steps are going to be initiation, elongation, and termination. And you should understand what's happening with the mRNA, the ribosome, and the tRNA at each step in that process. Once we have formed a polypeptide, we are going to send that to the rough endoplasmic reticulum, and that polypeptide will undergo post-translational modification. That's a big word, but it just means that it's going to happen after translation happens, so post-translational. And modification is just either editing or reshaping or adding extra things on. So proteins are going to have a little address label. It's a sequence of amino acids that says, send me to this location in the cell. Based on that address label, the rough ER can tell whether that protein is destined for secretion or packaging or some other fate within the cell. So proteins that are destined for secretion or packaging are assembled on the rough ER, and then they're going to look at that little address label and they're going to get sent to the Golgi complex. From the Golgi complex, they're either going to be prepared for secretion or packaged up in something like a lysosome. A signal recognition particle, that's the address label. It's going to drag the new protein from the ribosome through a pore into the cisterna, or the middle of the endoplasmic reticulum. This is where most of our post-translational modification occurs. Sometimes we remove some amino acids. We can fold up the protein by adding very strong disulfide bridges or adding carbohydrates to make it into a glycoprotein. The rough ER is then going to pinch off transport vesicles, which will fuse with and empty into the nearest Golgi complex for further packaging and secretion. So the signal recognition particle is the essential component of the new polypeptide that functions as an address label. So it tells it where to go. You can see in figure 3.3 from your textbook this sequence occurring. So as the polypeptide is produced, that signal recognition protein is going to help drag it into the rough ER. That rough ER is going to modify it as needed, and then it will send it off to the next step in the process. I would encourage you to take a further look at that figure to understand post-translational modification. So once the peptide or protein reaches the Golgi complex, it is going to be modified as needed within the Golgi complex. The middle portion of the Golgi complex, what we call inside the Golgi complex, is called the cisterna. So once it's modified, it's going to be passed to the next cisterna, and eventually we get all the way through the Golgi apparatus, and the last cisterna is going to release the finished product as a membrane-bound vesicle. So a vesicle that's surrounded by a piece of membrane. Secretory vesicles migrate to the plasma membrane and release their product by exocytosis, which we talked about in membrane transport. And vesicles which will remain in the cell are called lysosomes. And those vesicles just package up some of that protein, usually it's enzymes, and they just hold on to those enzymes within the lysosome. Lysosomes are important for breaking things down. So when that lysosome fuses with another vesicle and needs to destroy what's in that vesicle, those enzymes will be there to do that. We have one special structure which helps with breakdown of proteins. These are called proteasomes. They're barrel-shaped structures that destroy proteins that are unneeded, damaged, or faulty. These proteins that are damaged or faulty are tagged with a structure called a ubiquitin. So this ubiquitin focuses as a tag. It's a destroy tag. It says, hey, come destroy me. These proteasomes are able to cut long proteins into smaller peptides. By doing so, it destroys the function of the peptide and begins the process of recycling the amino acids. So these proteasomes are hollow barrels, no top, no bottom, and as the protein goes through that barrel, it gets clipped into smaller portions or peptides. This is figure 3.34, and this is a diagram of information transfer from DNA to RNA and then to the polypeptide. So this just reviews how triplets on DNA go to codons on mRNA, and those codons match up with anticodons on tRNA, and those tRNA molecules with the anticodons are bound to specific amino acids, and that's how we get our polypeptide amino acid sequence. This wraps up our discussion of transcription and translation, and next we're going to move on to cell division.